I'm Tim Ventura, and we're joined today by Dr. Clarice Aiello, Assistant Professor of Electrical and Computer Engineering at UCLA and Principal Investigator at UCLA's Quantum Biology Tech Lab. Clarice is a quantum engineer interested in how quantum physics informs biology at the nanoscale, and her research group works to perform quantum measurements on living sensors such as proteins, cells, and microorganisms to understand how they interact with their environment. Clarice has a PhD in electrical engineering from MIT, an engineering diploma from École Polytechnique, and a master's of philosophy and physics from the University of Cambridge. She's held postdoc appointments at Stanford and UC Berkeley and has expertise in quantum dots, photonic structures, and nanosensors harnessing room temperature quantum effects in noisy environments. So Clarice, welcome. Thank you so much for joining me today.
Well, thanks for having me here and giving me the opportunity to talk a little bit about quantum biology.
You know, when I saw this on your resume, I saw you on LinkedIn and I said, oh my God, this is an interview I have to do. This is really exciting. This is one of the ultimate emerging fields for the 21st century, and you are right on the cusp of it. So thank you so much for joining me. So today we are talking about quantum biology. Again, this is really exciting, emerging field of study. So I want to start out with some definitions for the audience. What is quantum biology, and how does this field differ from traditional biology or biochemistry that people might be more familiar with?
Okay, so it starts complicated, right? Because there is no agreed upon definition of what quantum biology is. I'm going to tell you what I think it is and also what I think it isn't.
Okay.
I like to define quantum biology as the study and the control of putative quantum mechanical degrees of freedom that exist endogenously in nature, okay, for the purposes of therapeutics and maybe even developing better technologies. Okay, we can explore a little bit more what I mean, why I said the word putatively exist in quantum biology, in nature and things like this. But let me before tell you what I do not think what quantum biology is, because there is a lot of name hacking around. I do not think quantum biology is using technological quantum sensors to study classical biology. I do not think quantum biology is using quantum computers and quantum algorithms to improve healthcare or healthcare to analyze healthcare data or to improve to do drug development. And most of all, I do not think quantum biology is related to new ageisms, right? It's not related to telepathy, how we talk to each other, and okay, that's all I wanted to say.
No, that's
Oh,
absolutely wonderful. I'm sorry, did you have more? Or
well, I have to explain why I said that quantum biology is the study of the putative quantum degrees of freedom that exist in biology, and that it is already a little bit intertwined with your question of how does this differ from classical biology and from biochemistry. So if you humor me, let me give you a little bit of introduction of what I do, because I'll get to how quantum biology I think is different from other disciplines, and why I am still cautious to use words such as quantum degrees of freedom that putatively exist in biology.
okay.
So my regular spiel on what I am and what I do is I like to call myself a quantum engineer. This means that me and my group, we develop instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So it can actually be proven, and that's my PhD, that's the field that I was during my PhD work. it can actually be mathematically rigorously proven that if you use a quantum object, such as an electron, as a sensor, your measurement is improved. In other words, the sensor quantumness enhances your measurement. This is the field that is known as the field of quantum sensing. There are now technological quantum sensors that exist and that give information about processes in general. For example, I believe that Schlumberger is using technological quantum sensors to prospect for oil. There are people developing better magnetic resonance imagers based on technological quantum sensors, etc. However, there is incomplete but compelling evidence that nature might be using a similar process to that of quantum sensing. For example, electrons in nature might be using a similar type of non-trivial quantum behavior in order for, say, the proteins where those electrons live, in order for that protein to interact with their and respond to their environments. Okay, so let me talk to you about this evidence. So in order to talk to you about this evidence, I will need to tell you a little bit about the flavor of quantum biology that I play with that is related to this quantum sensing. So this flavor of quantum biology has to do with the quantum property of spin. Okay, so spin is a fundamental property of matter that is as fundamental as, say, mass and charge. In the same way that mass measures how an object interacts with the gravitational field, right? Big mass or small mass. Charges measures how, say, an electron or a positively charged cation interacts with electric fields. Spin measures how well an object interacts with a magnetic field. Okay. So electrons have spin, like atomic nuclei have spin. And in the same way that charges, it's an abstraction, right? Charges are positive or negative. They could be called like A and B, whatever. Spins are usually represented with arrows, spin up or spin down, meaning different ways of interaction with a magnetic field. It has been known in chemistry for decades, there is no controversy in that there is a class of chemical reactions that depend on spin. So here's how this goes. There's a chemical reaction happening, and then at some point, the chemical reaction comes to a crossroads. At that point, the chemical reaction effectively looks for the spin state of a particular electron. If that particular spin state is up, the chemical reaction continues through one branch. If that particular electron spin state is down, the chemical reaction continues through another branch. Importantly, the final macroscopic products of those two branches are different. So there's no doubt that affinity quantum property of electrons is known to play a role in the macroscopic fate of a class of chemical reactions. Now, again, if for test tube chemistry, if at the moment where this chemical reaction is at this crossroads, the particular spin that we care about sees an external magnetic field, this electron spin will interact with this external magnetic field in pretty much the same way that electrons in technological quantum sensors interact with magnetic fields that say they sense. So this is a bona fide quantum sensing phenomena. And the result of that is that this electron spin is going to respond to this magnetic field. It's going to it may be altered by this magnetic field, so that the magnetic field can actually alter if the spin is up and the chemical reaction continues here, or if the spin is down and the chemical reaction continues here. So chemical reactions are known to depend on electron spins, and magnetic fields are known to change those spin-dependent chemical reactions. There's no doubt that this happens for test tube chemistry, except that people have been observing effects of magnetic fields on biological matter for more than 50 years, and a lot of those effects like how biological matter responds to magnetic fields, seem to be consistent with what would be expected were this type of spin-dependent chemical reaction happening inside proteins, inside cells. Okay? So this is somehow counterintuitive because quantum phenomena usually happens at very low temperatures in organized systems, right? And so it might come as a surprise that there might be responses to magnetic fields that are consistent with this type of spin phenomena.
Well, and if it's okay, I want to jump in and read
Yes.
something that you had written and hopefully put this in a little bit of context. I'm jumping around in my questions, but
Go for it.
you'd written that many disease markers, such as the production of reactive oxygen species during adult stem cell mediated regeneration, epigenetic changes to induce pluripotency, cell fate decisions in embryonic stem cells, cell differentiation of adult stem cells, were demonstrated to be controlled by weak magnetic fields. And this comes back to spin because you're talking about imagine driving cell activities to treat injuries and disease simply by using tailored magnetic fields.
Yes,
That's giant. That is giant.
That is giant. And the thing that prevents this giant leap is the absence of unambiguous evidence that those things that people see happening inside cells are actually confirmed to be driven by those spin phenomena, right? If this is confirmed, the cool thing is that, yes, you can drive, if you will, chemical reactions inside cells that might affect disease progression and stuff in an easy way, in a way that is endogenous, no need for genetic engineering, non-invasive, portable, cheap, right? It's electromagnetic medicine and accessible to anyone who has a mobile phone, right?
Yeah. Well, and again, if I could jump in, and this is why I think you wanted to separate it from kind of the new agey aspect of things, right? Because I've seen these for sale online. People will sell magnets. They say, put this magnet here and it will treat this. You're not talking about that. What you're talking about is something that's tailored, very tailored to change spin states, to change the actual chemical properties that lead to these reactions. So this is very, very detailed work.
That's very cool. And the idea is that, you know, we know from test tube chemistry that there are all those phenomena that like changes as a function of magnetic fields. What if we could bridge those two length scales between test tube chemistry and like cells and try to come up with, say, a code book on how to deterministically tweak the fate of chemical reactions inside cells, not by chance or by finding by magic fortuitously like a magnetic field that does something, but by design, because you understand nanoscopically the spin physics of what's going on, right? There are many, actually, I need to talk to you about, there's this device called Optune. It's now a company that is valued at $9 billion, they have found a magnetic field that treats glioblastoma, that is a form of brain cancer. They know that the device is the least important thing. The thing is that they found a field, a particular magnetic field intensity and frequency. I don't think they know nanoscopically what's going on. They know that the field messes up with cancer cell division, proliferation, in a way, again, that is consistent with this spin phenomenon. And imagine if you were able to select the type of ailment that you want to cure with your cell phone, right? Not going, well, we found this field haphazardly, but understanding nanoscopically what's going on so that, well, today you go there like, well, today I need help with wound healing. Wound healing is another thing that has been shown to depend on magnetic fields in a way consistent with spins. You say, today I have something here, I want help with wound healing. Then you go to your app and click wound healing, and then your crappy cell phone produces the exact magnetic field intensity and frequency that you need, and then you go there. This is science fiction nowadays, right? Apart from some FDA-approved devices such as Optune, and I think there is great potential in trying to understand this electromagnetic medicine in a rigorous way. Right?
Yeah. Well, I wanted to ask if any of this came out of MRI machines, right? Because with magnetic resonance imaging, you are aligning the spins, if I understand it correctly, the spins for all of these. And I thought, you know what, that must be one of the great demonstrators of how that affects chemistry.
No, and that's the coolest thing ever. Hear me out. The mechanism is very different. For reasons that are well understood, there is no mystery, no controversy at all. The magnetic fields that alter such electron spin dependent chemical reactions, they are relatively weak. They are on the order of your cell phones, on the order of the magnetic field of the Earth, that is like orders of magnitude smaller than the field that you sense when you put your cell phone close to your face. And that's extremely important because, for example, if you put the magnetosensitive proteins in a test tube, proteins that can sustain such spin-dependent chemical reactions inside an MRI machine, that big field is like the Tesla level field is not going to influence which chemical pathway is taken. And that's super important because this means that, well, that the good part of that is that you don't need a very intense magnetic field for those things so that it can be portable. This is the good thing, but it's also the bad thing because if someone bad learns how to tweak physiology in a bad way, it means that they wouldn't need big
Okay.
electromagnetic weapons that emitted a super high intensity either, right? So the key point is that the magnetic fields that influence this stuff is small, it's tiny, right? So it's easy to influence those things if only you knew like the Goldilocks region for each chemical reaction.
Okay. Well, if it's okay, I want to jump into a few other examples of quantum biology for the audience.
Yes.
And I did a little bit of research before the interview. Again, I'm just trying to provide context and give people something that's a little familiar to them. The
Yes.
best known, this is one that I've read about in the past, maybe photosynthesis in plants,
Yes.
and where the plant absorbs light through the process of electron excitation in little microscopic antenna structures. So if I understand correctly, this is kind of where the field began, right?
Um, there were several different branches of quantum biology developing at the same time. I can talk about spin again in a second and how the
Oh,
spin
yeah.
part started, but let us talk about photosynthesis. And again, there is all the studies for photosynthesis have been done for things, say, in solution, in very controlled environments. To this day, they're in the same way that for the spin flavor, to this day, there is no unambiguous proof or refutal that actually that's what's going on inside a plant, right? That's the leap of faith that needs to happen experimentally at some point soon. This said, yes, for um, plant structures in solution, there is evidence that quantum mechanics might be at play. Even though the traditional evidence for quantum-nesing photosynthesis is exactly what you just described. So the idea is that there's a photon absorbed here, and the energy of this photon needs to go to someplace else. Initially, people postulated that the carrier of energy that went from here to here, that this carrier of information called an exciton could actually explore multiple paths at the same time. And this multiplicity of paths, this superposition of paths, would make it very likely to reach the place where it dumps the energy and makes it reach this place optimally. So this is the standard view. There's a more, this theory remains, or this interpretation of results remain a bit controversial. There is a slightly more modern view of that process that also includes quantum, but doesn't include superposition of pathways, at least not predominantly. There is a more modern view that posits that what's helping, well, because photosynthesis is way more efficient than any solar cell. Solar cells are also doing the same thing. They're absorbing energy, and then excitons have to be transported into a place where they dump the energy. Photosynthesis is way more efficient than any solar cell made by humankind. So what might be helping this transport of energy be so efficient? The more modern view is that actually this process of transport of energy is actually helped, added by noise. People call it noise-assisted processes or thermally assisted processes. This means that things that people usually consider bad in quantum mechanics, say the presence of thermal energies that make things vibrate, might actually be making it easier for the exciton to go from one place to the other. This is
Okay.
very different from this like multiple paths. It's another type of flavor of quantum biology. I
Yeah.
think there's more and more evidence that this process is rather noise assisted than just like a superposition of paths.
Well, and then not all quantum biological processes are good either. I'd also read that the load-in DNA mutation model supports the idea that DNA nucleotide mutation might be happening through a process of quantum tunneling, right? So, and really, when I read about that, the thought that came to mind is, ultimately quantum mechanics is at play underneath all of this biochemistry. And so it makes sense that we would see quantum processes pop up here and here and here and here, including negative things like DNA damage, right?
Yeah, so just for the DNA stuff, there's beautiful theory done at that. There is no experiment, I don't think even in test tube, that unambiguously proves that there's tunneling going on. This said, let me tell you the good side about tunneling. Okay, enzyme functioning, the things that catalyze the things in your body, enzymatic functioning has been shown again in test tube chemistry to be absolutely dependent on tunneling, both of electrons and of protons. So tunneling can also be a good thing. But for all of that, for DNA, for tunneling as as pertaining to DNA mutations, and for tunneling pertaining to enzymatic functioning, and for tunneling pertaining to electron transport too, I do not think there is any conclusive data, say in living cells, that's happening. There's all evidence available in test tube chemistry, and there is no, if you will, quantum wet or measure or a quantum wet measurement, or a measurement that a quantum physicist would be happy with. And that I think explains a little bit the resistance of the mainstream quantum community in accepting.
Okay. Well, and the other one that I had read about, I think this is my final example, was quantum vision, which is a very complex term for basically just the cells in the eye relying on quantized energy to convert light into electrical impulses. So what was interesting about that, though, when I read about this, they mentioned the dark adapted human eye, and I've heard about this before. Back in the old days, I've heard that scientists would actually dark adapt their eyes for up to an hour to be able to see these low light processes, and that the eye is actually sensitive enough when it's fully dark adapted to detect a single photon. So that was just kind of a blast from the past, but really exciting.
So there are several levels of quantumness. I'll get to quantum vision in two seconds. Well, first of all, everything is quantum because everything is made of atoms,
Yeah.
right? That's very trivial. Everything that is quantum usually has quantized energy levels. This means that there are only certain energies that can be absorbed, which, for example, there would only be certain photons of certain energies that could be absorbed by a photoreceptor in the eye, which corresponds to a photon that can, say, make an electron be excited by a finite and well-regulated chunk of energy. So this is at the quantum level of what I would say quantum vision. I do not call quantum vision, people are going to kill me. I do not consider quantum vision super quantum biology because it's almost trivial quantum biology.
No, that makes sense. When I read that, I thought the same thing. I said, well, basically it sounds like they're relying on the quantum aspect of a pretty well understood process.
Yes. I think that where it gets interesting is when we talk about the next level of quantum, which is superposition, which is like one single quantum object being able to be in two positions at the same time, in two spin states at the same time. I think superpositions have never been able to be measured in proteins inside cells or inside a living stuff. Nobody knows if cells can sustain superpositions before they die out, because everything that starts quantum dies classical. Nobody has observed whether superpositions can be maintained, let alone utilized by cells, right? This is the level that would be needed for the spin stuff that I just described. It's a very
Okay.
basic level. Actually, that's all that is needed for technological quantum sensors to work too. It's like the superposition of states for a single quantum object. The final level of quantumness is what people call entanglement. Entanglement is a particular interaction between more than one quantum object. Okay, so if you do not have superposition, you cannot have entanglement, right? And as of now, I do not think there is enough evidence that nature, like even consistent evidence, like test tube evidence, I am not convinced that nature is using entanglement to function. I am ready to bet my career because of the type of experiments that we're doing. I'm ready to bet that nature is using superposition to function, but I'm not ready to bet that nature is using entanglement to function yet.
Well, if it's okay, let me jump into ORC-OR theory. And this is a little bit more speculative, but this is something that I've read about in the past. Physicist Roger Penrose and anesthesiologist Stuart Hameroff developed a model of consciousness. They're calling it ORC-OR, which I think stands for orchestrated object reduction. And it relies, they believe, on quantum effects in microtubules inside of neurons. As a mechanism for information processing. And I believe they've even speculated maybe non-local influence of some kind. Do you
entanglement, namely entanglement?
Yeah. Now, do you think that was what intrigued me about that was, and I believe Stuart Hameroff had said this. He'd said potentially this, if there is quantum processing going on in the brain somehow, that may give us orders of magnitude more processing ability than I guess we give the brain credit for.
Okay, let me preface everything that I'm going to say by saying that I respect Sir Roger Penrose immensely. And I think that their theory remains and will remain for a good time untestable.
Okay.
I put that theory in the same box that I put, say, string theory. Okay. So here's the thing. There's nothing fundamentally special about microtubules. Microtubules are very stiff biomaterial. There are other similarly stiff biomaterials. People have people have shown that excitons in microtubules can be like delocalized and things like this, which is good science. And I do not see the direct relevance to this theory. Again, right now in the world, there is no experiment that can show that a living cell is using superposition to function, right? And now you start talking about entanglement. I mean, if you cannot prove superposition, forget about entanglement.
Okay.
So,
well, no, I think that answers my question, and but I think the other thing that's definitely worth pointing out is they definitely put this forward as a hypothesis and it requires testing. And
Yes.
you know this on so much more of a granular level, right? This is what you actually work with on a daily
Yes.
basis. So,
Yes.
yeah, I think that addresses it very fairly. It'll remain in the realm of theory
For the time being, for the foreseeable future.
for the time being. Yeah. Well, so, I should touch on your hopes in terms of potential treatment for diseases, because ultimately that comes back to, I think, what your focus is with the research.
Yes.
What do you think you might be able to achieve? You mentioned, I think, a type of cancer that was treated with this. Do you think that we might be able to see more of that in the future?
Yes, and again, the technology needed to implement that, to get devices that produces, say, magnetic fields, the technology is there. You don't need anything else. What you need is a code book on how to tweak those electromagnetic. And I think there is a lot of room for electromagnetic. It's really like electromagnetic medicine. Don't if you Google quantum medicine, it's bad. There's going to be a lot of like new age stuff. Don't Google quantum medicine, but that's exactly what it is. It's quantum healing in a responsible way, right? We want to be able to just tap on those endogenous quantum knobs and see where we can take biology in a deterministic way. So I think this is just the start, right? Electromagnetics in biology is a taboo in the West in a way that it has never been in the East, in Russia, China, Japan. There's research of electromagnetic, so spin-dependent chemical reactions as applied to, and this is what I wanted to tell you, spin-dependent chemical reactions as applied to biology. I find papers from Russia from like the 70s on that, right? It's amazing. People were thinking about that. Uh, and in the West, I think there's a little bit more because of this new agey thinking. I think there's a little bit more of reluctance to bring this back into the mainstream, right? So, and that's what we're trying to do here.
Wonderful. Well, Clarice, again, let me thank you so much for your time today. And let me close, if possible, by asking, what do you see happening through the remainder of this year? What are your goals through 2023 and into 2024?
Um, I hope that quantum biology gets more and more recognized as a responsible field of research. There's a lot of stigma related to quantum biology, exactly for the reasons that we've been
Yeah.
describing, right? And I think that scientists themselves, I hope they get a little bit less dogmatic in the sense that like physicists sometimes won't want to engage because they're like, quantum effects at room temperature, right? And biology sometimes, if I say, well, you may not need to take a chemical, you might do this electronically. They don't get it either. So I hope that the scientific community in general uh, gets a little bit more tuned to this possibility, and I hope to keep expanding uh, this community. For example, we have just had the first ever Gordon conference on quantum biology. Gordon Research Conferences is like a series of prestigious conferences on many themes. And for the first time, they accepted our proposition to have a quantum biology themed conference, which was very big. I think it's a legitimizing step. And the conference was co-chaired by Young Chen Kim in Surrey and myself. So I think that was good. Uh, we have been having since the pandemic weekly online what we call big quantum biology meetings where we bring speakers who most of the times do not even identify themselves as quantum biologists, but it's people from physics, from biologists, just to talk to people who want to bridge those disciplines, right? And this happens. So my goal is to legitimize this field and to be able to like convince the broad public and the scientists that there's something there. There's something there. There's something there that is legitimate and that should be legitimately pursued scientifically.
Wonderful, wonderful. Clarice, thank you again so much for your time today.
I had fun talking to you. Thank you, Tim, for having me, and looking forward to interacting more.